Hello, I'm Dongyoung Kim, currently a senior at Arjun University. I'm going to be talking about my study focused on the Korean endemic harvestmen, Kaolinonychus, in an integrative taxonomic way. It is well known why cryptic diversity is important and how it contributes to ecology, evolutionary biology, and conservative biology. Cryptic diversity means the occurrence of multiple distinct evolutionary lineages that are hardly detected due to their morphological similarity. In particular, these cave or subterranean organisms are noteworthy from this point of view, because caves are isolated ecosystems causing frequent geographic isolation and speciation, simultaneously driving convergent evolution under the spatial and similar selection pressure of caves like a aquatic environment. Due to this unique combination of features, caves and subterranean organisms are supposed to harbor many cryptic diversities. And more cryptic diversity is being discovered with the introduction of molecular genetics and several up-to-date sciences. Next, let me briefly introduce the subject of my study, Opiliones. These somewhat spider-like organisms commonly called harvestmen or members of Arachnida. Despite their similar appearance, they are not actually spiders, but close taxon to spiders, mites, and scorpions. The order of Opiliones is divided into 4 suborders, and here a suborder Lineatres, which have extraordinary appearance, are of particular interest to me. Among the Lineatres, the family Paranonychidae is a cryophilic taxon with an amphibaryngean distribution like this. Although they are geographically far apart, they show highly conservative morphology led by strong ecological constraints. They are small predators and scavengers inhabiting microhabitats and are found in caves and under the log and litter. Members of Paranonychidae are known as dispersal limited organisms having high regional endemism and low vagility. And one of the family here is the genus Kaolinonychus, endemic harvestmen found throughout South Korea. There is one monotypic species, K. koreanus, with 2 subspecies, K.C. koreanus and K.C. longipes. They were what I'm interested in. According to previous study in another member of Paranonychidae, Squalobunus, morphology in reproductive organs does not sufficiently reflect their high genetic diversity. After I read this, I examined the genital morphology of Kaolinonychus, and I was able to get similar result, conservative genital morphology among the geographic groups. For this reason, I guess only a single species with broad distribution was historically regarded as a classification of Kaolinonychus under the traditional alpha taxonomic system. However, when observing them, they look different somehow, but it is not easy to figure out which part is different and set some qualitative criteria. So how should we approach this cryptic diversity? For this, an integrative taxonomic approach was used for deeper investigation, referring to the study of Jarkarbedian and Hedin in 2014 as a reference. I conducted analysis using mitochondrial CO1 gene sequence and morphometrics data with 65 length variables. And using them, I conducted integrative taxonomic analysis. And now here is the result. First, in the result of phylogenetic analysis of the mitochondrial CO1 gene, we can see 3 major clades, and in detail, there are 6 subclades. I set these 6 subclades as pyrrhic groups in analysis because these clades are identical in both maximal likelihood and Bayesian tree, excluding one exceptional group with Tebek and Mountain Bagun clade, a polytomic group in Bayesian inference. But initially, I set these groups as pyrrhic groups, subject to analysis I conducted after it. Okay, now there are 6 subclades, and this bar means the classification of Seicho Suzuki, 1 species and 2 subspecies. And these are the appearance of each type. We can observe slight differences among each other. First of all, I used some statistical support values like bootstrap or posterior probability of in phylogenetics of each clade as threshold of delimitation. In this process, I set 70% and 90% as the specific threshold values in each maximum likelihood and Bayesian inference, which are generally accepted. From this, we can see that three clades, which are denoted as A, B, and C, are highly supported, and the inner clades of the C clade, colored with type of yellow, are weakly supported except for the Northern Gangwon and Gyeonggi clades in the Bayesian tree. So I could represent this result like this. And then I conducted dimensionality reduction of morphometrics data. This is the result of principal component analysis. Look at this, it can be seen that the A clade and B clade are very close together while forming their own clusters. And in the case of the C clade, a narrow cluster did not appear, and there was no cohesiveness within each subclade in clade C. So I can express it like this. Next, in PAM clustering, partitioning around meadows, 2 to 4 clusters were found to be suitable, and the clustering results for each K were shown as the following. You can see that clade A and clade B are separated from clade C as K increased, although the symbol K population in clade C, shown in green, is exceptionally isolated individual. As such, it was possible to establish this final species hypothesis by synthesizing the result of cladistic analysis of genetic data and panatic analysis of morphological data. According to the hypothesis, the three major clades are estimated as 3 individual species. In the validation process, first of all, pairwise FST was calculated to estimate the degree of genetic differentiation between subclades. As a result, the red clade displayed above 0.75 from all the other clades. The blue clade showed over 0.5, while 4 yellow type groups showed around 0.4 or below among each other. Next, the t-SNE clustering using morphometrics distinguished the 3 clusters as separate groups. And this is the result of partial risk scale discriminant analysis in PRSDA. In PRSDA, the dimensionality reduction proceeds in the direction of discriminating the data labeled into 3 groups by reflecting the hypothesis established above. Looking at the result, the 3 clades are well grouped together, and let's take a look at the contribution of variable, which is the main purpose of PRSDA, for selecting the feature variables that highly contributed to their distinction. As will be discussed in more detail later, most of the walking leg length ratio is ranked at the top. Next, ABGD and BPTP algorithms were used as a molecular operation taxonomic unit, most of which distinguished much more species than our hypothesis. This seems to be due to high genetic endemism in Paranonychidae. Nevertheless, since these subgroups were not grouped across the hypothesis, so it was judged that our hypothesis was supported. And finally, I concluded that these 3 major clades could be determined as separate independent species. Among these 3 groups, C and A, yellow and red groups, were originally classified as 2 different subspecies by Seishu Suzuki. Each is clade corresponding to KC coreanus and KC longipes respectively. My argument is that these existing subspecies are clearly both genetically and morphologically different, so they should be elevated to individual species. And I'd like to say this blue clade could be a potential new species have never been recorded before. But however, I believe that an advanced investigation, including more thorough sampling, is required for it. Beyond the limitation, I tried to quantitatively analyze what traits differ and what morphological traits can effectively distinguish them. The basic way to do this may be by inspecting the loading values in the PCA. Especially in our PCA research, this can provide informative information because the three groups were classified without overlap in the course of principal component 1. In addition, we were able to conduct feature selection using PRSDA, a tool principally used in the metabolomics field. A feature selection is finding the variables that contributed greatly to dividing two different groups. So in taxonomy, it can be very similar analysis to finding the key characteristics. Using the variables selected in the PRSDA, we drew a box plot for each group and conducted an ANOVA test. The results showed that walking legs were clearly high in A clade. And the thickness of the appendage segment, fatty pulp thickness, and body ratio were relatively high in clade C, identical to KC Coreanus. To sum up, I could conclude that the three clades within the genus Kaolinonychus are presumed to be individual species that have experienced independent evolutionary history, especially those that have been originally divided into different subspecies. KC Coreanus and KC longipes should be elevated to species level. In addition, key characteristics were also found between the two species with statistically significant difference in length ratio of the appendages and appendage segment thickness between the two clades A and C. One remarkable thing I experienced during this study is that the decisive difference between them, the ratio of second walking leg, is exactly the same as Suzuki's suggestion. In my study, I found out which traits were important in a quantitative way based on statistical methods using the characteristic data that I took as many as I could measure. And the result was the same as the key character that Suzuki suggested more than 60 years ago. Although there are differences in the detailed criteria of delimitation, I was able to admire the amazing insights of former taxonomists in spite of the technology of the time. What I think more valuable part of this study is the rediscovery of KC longipes. There has been no record of this subspecies since 1967, and at least no species have been left since 1967, as far as I know. I went to the Gongi Cave, which is the type locality and only known habitat. But when I visited myself, I could see the cave illegally occupied and renovated by shamans was thought to be unable to function as a natural cave anymore. The devastation was terrible, really terrible. The floors and walls were cemented and the ceiling was covered with urethane and artificial lighting was installed inside. So I couldn't expect the harvestmen to live there anymore. And just as I was about to give up, I found a pile of stones with a small lens at the bottom of the atoll where I was able to collect unmatured KC longipes. I tried to collect a mature sample, but it was impossible because the shaman came back to the cave and he expelled us. Nevertheless, fortunately, we were able to investigate another cave in a similar environment near the Gongi Cave, where we were able to capture a number of mature samples similar to KC longipes. And according to the DNA barcoding analysis, we found that the unmatured sample of Gongi Cave and mature samples from the neighbor cave matched almost 100% on the CO1 sequence. So we've confirmed that they can be grouped into the at least the same species. I think conservative biological research is necessary for them, considering that their habitat distribution is very limited and they are quite understudied. And as I showed, even the limited locality has been severely damaged. Although it has been difficult to receive conservative management because they have been divided into subspecies level, not a species level so far. But it is expected that more proper conservative approach and management will be possible if they are elevated to species level. Next, in the case of bee clade, they are expected to be the candidate for new species, but it is necessary to further study whether it is clearly different from the yellow-colored sea clade through more thorough sampling near the karst region of Gangwon-do, where they were collected. Finally, in the case of sea clade, same as case Koreanus, a complex relationship with several clades are expected within it. Therefore, it is necessary to study their evolution at the population level. And I think this would be a very interesting topic. So I think we need to conduct advanced research on the microevolutionary approach. This study is thought to have rendered meaningful conclusion in that we discovered the cryptic diversity of the harvestmen that has been hidden in a veil for a long time. And for this, we gathered the multiple data and conducted an integrative taxonomic study through various quantitative analysis. I expect that the method used in this study will also be applied to the detection of cryptic diversity of various subterranean and cave organisms. Thanks for listening.